The chrome alveolates are thought to have arisen from a common ancestor that engulfed a red algae. So that would have been the primary endosymbiont. So all of these can be traced back to that common ancestor point. Now, when we talk about them, we've got several groups within the chrome alveolates, and one of the biggest groups is the alveolates themselves. And these are known for having membrane-bound sacs. And so these little sacs are found underneath the plasma membrane, and that's what you're seeing these little white arrows point to. These sacs have numerous functions, and they're thought to help support just the general structure or stabilization of the surface of these cells, and they may also be involved in regulating water content or ion content within the cells. There's three main groups of alveolates that we're going to discuss. And so those are the dinoflagellates, the apicomplexans, and then also the ciliates. So as we talk about these, keep in mind that each one of them is going to be having these alveolate cells, sacs inside of them. So the first ones are the dinoflagellates. And the dinoflagellates themselves have kind of a unique look to them because these have this armor that kind of makes up the outside of them, or that's what they look like. And this armor is made up of cellulose. So they have cellulose plates that surround them. And then another thing about them is that they have these little flagella, which you're seeing here. They have flagella that run down the middle of the plates. Down the middle or between the plates. Another thing about these dinoflagellates is that when they're moving through the water, they have a spinning movement. So that's a very unique kind of look to them. We're talking about aquatic organisms here. These are going to be a major component of plankton. So these are going to be photosynthetic organisms. They're going to be found in a lot of plankton. And when they become too abundant in the oceans, this is when we have the red tide events. So when you hear about red tide, what that's referring to is basically a lot of dinoflagellates in the water. And these happen to have a red pigment inside of them. It's a carotenoid pigment, and that produces that red coloration, which is what the name is from, red tide. The problem with this is that dinoflagellates also produce some toxins which cause a lot of kill-off for marine species. So this is definitely not a good thing when we're talking about red tide events, but without the overgrowth of them, they are a very good source of plankton for marine animals. The apicomplexans, these are known for their apex, or what's called the apex. Most of these are going to be parasites, and that apex is going to be for penetrating another cell or penetrating the host organisms. So what we're talking about is this little part right here. So this is unique, and it has organelles that specialize in penetration. So these will be very good at basically inserting themselves into that host organism when they come into contact with them. These also have a modified plasmid, which is called an apicoplast. They many times have unique life cycles such that they're parasites, but these parasites might require two hosts. We're going to look at one in just a moment, and it's the one that causes malaria, and it can't just survive in humans, it can't just survive in mosquitoes. It has to complete the full life cycle using both host organisms. These tend to spread as what we call, put it right here, sporozoites. Okay, so these will be special little cells that have that apex that are going to be very good at inserting into the host. So if we look at this slide here, this is showing Plasmodium. And Plasmodium is the protist that causes malaria. So we're looking at the life cycle of this organism on this slide. So what you're seeing up here is the very beginning of it, where that mosquito that carries the Plasmodium protist feeds on a human, and that Plasmodium protist now enters the bloodstream of the human. And it's entering as the sporozoites that we mentioned on the previous slide. Those travel to the liver, they infect liver cells, and once there, they're going to multiply by the process of mitosis. This produces a lot of additional cells for the Plasmodium, and these are going to basically travel around with the red blood cells. They're going to insert into the red blood cells because they're able to do that because of that nice apex that they have. And then they're going to form some gametocytes, which will develop further into gametes. Now down here at the bottom, we have another mosquito that is feasting, and he will be taking in Plasmodium in the form of the gametocytes. So here we have the start of the other half of the cycle, which is going to be within the mosquito itself. So within the mosquito, those gametocytes will develop into gametes, and then those gametes will have male and female ones, and those two will be fertilized and will get the zygote formed. That zygote will then develop into the sporozoites, which you're seeing here, and then we have basically run the life cycle all the way around. So now if we have a mosquito that's carrying around the sporozoites, if it feasts or feeds on another human being, it's going to transmit that malaria to the next human being. So again, these are the apicomplexans. Notice that they do take two hosts a lot of times. This is fairly common for these. The vast majority of them are going to be parasites. The last group of the abiolates are the ciliates. And the ciliates, you might have noted from the name ciliate, they're going to have a lot of cilia. So these are short hairs that you would find all over the surface of these cells. And you can see that in the picture here that we've got lots of little hairs on the surface. These are used for movement. They can be also used for feeding. So it can create a little current to help bring things towards it for feeding. Another unique thing about these is that they have two nuclei types. So they have the micronucleus, and then they also have a macronucleus. So with these two nuclei, they are going to perform binary fission, and they're also going to exchange micronuclei with other ciliates through the process of conjugation. Here we see a look at a typical conjugation event in these ciliates. We'll have two sexually compatible cells that will come together and they will attach to each other, and at this point they each have their own diploid micronucleus. That micronucleus is going to undergo meiosis so that you end up with 4 haploid micronuclei inside each of these cells. What's then going to happen is that 3 of these in each one of the cells is going to disintegrate. So those are going to go away, and then the remaining one is going to divide by mitosis so that ultimately we end up with 2 genetically identical micronuclei that are haploid in each one of those cells. The next step is where they actually exchange genetic material with each other. So they will pass one of those micronuclei to the other cell so that each one of the cells has one of each of the micronuclei. Then the next step is that you're going to have fusion. Those 2 genetically different haploid micronuclei are going to fuse with each other so that we end up with a diploid micronucleus. So this one cell there is going to be diploid, and that diploid micronucleus is then going to undergo several rounds of mitosis so that ultimately you end up with 8 of these. Okay, so here we see 8 micronuclei. At that point, what happens is that they grow, 4 of them are going to grow into a macronucleus. And that's where you see them actually get bigger here. So they're going to actually develop individually into one of these macronuclei. 4 of them will do that. So we end up with one cell here which has 4 macronuclei and 4 micronuclei, and then we have just binary fission happen here. So this is where we have the binary fission, and that will then give us 4 identical cells there from the one original that we started with. The second big grouping of the chromoaviolates is the straminophiles. And what these are known for is what you're seeing in this picture here. They have two flagella. One of them happens to be hairy, one of them happens to be smooth. So that's kind of their defining characteristic. If we look at the individual groups within the straminophiles, the first one that we're going to talk about are the diatoms. Diatoms are going to be unicellular. And these are real pretty little cells when you look at them in a microscope because they look very glass-like. And the reason they look very glass-like is that they have a lot of silica in their cell walls. And not only do they have silica there, but they have this box. It's like a top and a bottom that kind of fits around their cell. So they have this silica box that provides this outer covering for them. It overlaps. And these are going to be a major component of plankton. These are found in huge quantities on the ocean floor. Because of all that silica, they don't decompose very readily. So this is used a lot as when you hear the word diatomaceous. Trying to get the spelling here. Diatomaceous earth. If you've heard that term before, that is the remnants of diatoms. So those are little bodies of diatoms that have been collected. This is a good medium for filtering. And it's also good for a soil conditioner a lot of times. People will add it to their soil to help replenish some of the organic material in their soil and also to just add some porosity to their soil. So those are the diatoms. Another one that is part of this group is the golden algae. In the golden algae, we have some forms that are unicellular or single cells. And then we also have some that have more of a colony life cycle or lifestyle. And that's what you're seeing in this picture here, is you have multiple individual cells that are living together, working together as a colony. So they're working kind of towards a similar goal here. The golden algae have brown and yellow pigments. And that's where the name comes from. These are going to be able to photosynthesize, and they are typically biflagellated, which we said is kind of a major characteristic of this group of straminophiles anyway. Okay, so those are our golden algae. The last of the chromoaviolates that's considered an algae would be the brown algae. And the brown algae are the largest of all of the algae. These can be hundreds of meters long. So these make up the giant sea kelp. So here you're starting to see some of the wide range of sizes that we have when we're talking about the protist. The brown algae have brown pigments, okay, which can give them the color for their name. Can also cause them to look kind of red or dark red. So it's not always The brown algae are very large algae. So this is one of the very large or the largest protists that we know of. These make up the giant sea kelp that are found in the ocean. The brown algae have a lot of brown and olive colored pigments. So that's the color that you tend to see them. These have kind of a plant-like structure to them. And by plant-like, they have features that are similar to the plant. So they'll have structures that look like leaves, that look like roots. We don't call them leaves and we don't call them roots because when you get down to it, they do have a different makeup to them. But we'll look at that overall structure in just a moment. So these are mostly marine. So they live in the ocean and they're going to be all multicellular. And not just multicellular, but very, very large. So if we look at the general structure of these, this is what we have. It looks like a small plant when you look at it in first glance. It has what looks like a stem, and I don't know if you can read it here, but that's the stipe. So the stipe is like a stem. It has what look like leaves, and those are the blades. And then it also has what we call a holdfast. The holdfast would be similar to the root. Now the difference here is that the holdfast is really just for anchoring purposes. It's going to hold it in place. It's not going to absorb nutrients or perform any of those functions that we tend to think of when we're thinking about roots. The one other thing is that it has these little bladders or air bladders. And these air bladders are going to help the plant to stand upright in the ocean. So these are very, very tall plants. These will help them to float towards the surface, which is going to be very important for these because these are photosynthetic cells. So they need to be able to capture as much light energy as they possibly can, and being closer to the surface is going to help them do that. Here we have the life cycle of the brown algae. This is also a very complex life cycle. We start to see some of what we call alternation of generations. And that alternation of generations refers to the fact that we can have multicellular haploid organisms and multicellular diploid organisms. And this is something that we don't see when we're talking about some of those other protists, or when we talk about some animal cells, fungi. It's something that's fairly unique to plants, but we do see it in the brown algae and a few other organisms. So what we're looking at here is that within the blades, you do have some cells that are diploid that we call sporangia, or they develop into sporangia. And what the sporangia do is they produce spores through the process of meiosis. Once we get those haploid spores, we call them zoospores. And those zoospores can germinate or grow into the gametophyte. That gametophyte is going to be multicellular, and we have female gametophytes, which will eventually produce the eggs, and we have male gametophytes, which will eventually produce the sperm. Once we do have the egg and the sperm produced, those two will come together. So the sperm is going to be flagellated, so it will be able to swim to the egg. And then at that point, we will have fertilization. So we get our zygote, which is our first diploid cell. And that diploid cell can then develop into the full-fledged sporophyte, which is the plant-like structure that we were just looking at. So notice on this, when we talk about the alternation of generations, notice that down here, we have multicellular gametophytes, so more than one cell there. And we also up here have our multicellular sporophyte. So that's what we mean by that term, alternation of generations. The last group of the straminophiles is the oomycetes. And the oomycetes are kind of an odd little group because for a long time they were thought to be part of the fungi. It's now known that they're quite different from the fungi, but they do tend to resemble them. So that's a case of what we would call convergent evolution. They've been subjected to the same kind of selection pressures, and over time they've learned to kind of deal with the selection pressures in the same way, and they've developed the same general structure, which is going to be this filamentous structure. So these are filamentous, and that's what's being shown in this picture here. When we say filamentous, they have what are called hyphae. Hyphae are those long strings of cells hooked together. And what that will do is it will maximize surface area. So these are going to have a maximized surface area, which will allow them to better gather nutrients around them, better gather water around them. It's just really good for their nutritional benefit, help them to get more of everything that's from their environment. Another thing about these that makes them, that is one reason why we do consider them different from fungi, is that their cell walls are made of cellulose. We'll talk about fungi in a later chapter, but fungi cell walls are all made of chitin. So that's a very different material from the cellulose that's making up the cell walls here. These are primarily going to be decomposers. We do have some that are parasites. So the water molds and the relatives of the water molds would be the parasite ones. And they do have kind of a unique life cycle. So if we look at that here, we have the hyphae, which are the these are the filamentous strands, and this is the diploid part that we're looking at here. And within those, we have certain cells which develop into what we call a sporangium. That sporangium produces zoospores, and those zoospores grow into other sporophytes. And so that would be the asexual form of the life cycle. We can also have them actually undergo meiosis. And when they undergo meiosis, they're going to produce an oogonium, which will produce the egg. And then we'll also have some male versions which produce the sperm. And now that you have the sperm and the egg, those can go through the process of fertilization, and we can get a zygote that develops. This structure that you're seeing, that oogonium, is fairly unique. Okay, so here would be a picture of the oogonium. Here you can see the individual eggs inside, and that's a very distinctive structure if you're looking at these oomycetes in a microscope.